So in this first lecture, we're going to start exploring different aspects of the immune system. And you'll notice in the textbook that the detail is fairly general at this point. We're sort of going to get an overall view of different aspects of the immune system and some of the cells that are involved in immune system function. And we're going to get it at an introductory level first. Later on, we're going to be adding a huge amount of detail. Now, immunology is actually a somewhat challenging topic to teach because it has so much detail potentially. We're going to try to strike that balance between general themes and general knowledge, things that you will actually carry with you out of this course, things that you'll actually remember. And then also we're going to have enough detail so that you can sort of back up that general knowledge with enough detail to make it meaningful. But what we're not going to do in this course is we're not going to drill down and try to memorize huge amounts of detail at the really fine level. I don't find that particularly useful, especially because it's not something that you're going to carry with you later after you're done the course. I want you to have a solid solid footing, a solid knowledge base, if I can talk here. And then I want you to be able to use that solid knowledge base and then as you need it later on to go look up details to plug into that. So it's kind of like big picture is always the most important thing for me, and then details can be added later or looked up later. A lot of the really nitty-gritty detail for immunology I don't actually know off the top of my head. I have to look it up just like everybody else. But I have that broad knowledge to know where the pieces fit, and if I need to look up a specific enzyme or a specific receptor, then I can do that by grabbing a textbook just like you're going to do in the future anyway. So that's kind of the goal. We'll talk a bit more about that as we go through, sort of what that balance is between detail and big picture. But as you know, if you've had a class with me before, I am always a big picture person first. If you don't have the big picture, the details don't matter. You need that context to fit those details into. So when you're studying for exams, for example, always get that big picture. Of course, I'll give you review questions that are very much directed towards that big picture, and then you have enough detail tucked away in your memory bank that you can use that detail to illustrate the bigger picture. Okay, that's a bit long-winded, but that sort of gives you a little bit of my teaching philosophy if you've not had a course with me before. So let's start with this overview of what immune systems are. The first thing I'll say is that this course tends to be very human-centric, and that's okay. We're interested in humans, we're interested in our immune systems. But it's a mistake to think that only humans have immune systems. Immune systems are very widespread and they're very ancient. They've been around for a long time. We're also used to thinking about immune systems as adaptive immune systems because we happen to have an adaptive immune system. But that's something that's specific to one branch of the tree of life, to jawed vertebrates, where of course we are of course part of that group. But that's not to say that other organisms don't have immune systems. They just don't happen to have adaptive ones. And we'll talk a little bit more pretty soon about what adaptive versus innate is all about. Immunology is also historically a very successful discipline. It's had very practical benefits for humanity as a whole. If you could take a time machine and go back, a couple hundred years to a world that didn't have vaccines, for example, people died routinely from all sorts of different things that we don't even really think about now, or are not, non-issues. Smallpox, for example, this one example that the textbook uses. It's actually one of our best success stories in the immunological world because we've managed to eradicate this disease, which was a horrible disease. Lots of people died from it. Many more people were disfigured by it. It's a viral disease, and because its reservoir was uniquely human, it doesn't have an animal reservoir, as it were. There's not other species that can propagate it. It made it a very good target for potentially eradicating it through the use of a vaccine. If you can vaccinate enough people, then eventually the herd immunity or the group immunity of that vaccinated group doesn't allow for transmission from one individual to another. So nowadays, in the present day, you are most likely not vaccinated against smallpox. If you have a smallpox vaccination, you'll have probably on your shoulder that sort of little donut-like shaped scar that results from a smallpox vaccination. I'm actually old enough to have one. I should have been vaccinated against smallpox, but I wasn't because my mom at the time had some concerns about a skin condition I had at the time, and she was worried that that might somehow cause a problem with that. My brother has been vaccinated against it. It's not like she's anti-vax. And because that vaccination program was so successful, and it broke the chain of transmission, eventually that particular virus was eradicated back in the 1970s. So I have had students that have that vaccination present, because in some parts of the world that vaccination continued longer than it did in North America. But there's an example of a successful attempt, or a successful accomplishment of actually eradicating a virus through vaccination. So a true immunological success story. The next one that we're trying for is polio. We're actually getting relatively close. Polio in the wild is actually quite rare right now. The last couple of places that have it are places in Pakistan and Afghanistan, I believe. There's still a few places, and there's a few pockets in Africa as well. And the goal is that if we can vaccinate enough people in those areas, we can break those last sort of wild reservoirs of human-to-human transmission, and we can eradicate it. And the reason for targeting that virus is, again, it doesn't have an animal reservoir. So there's not an animal population that will keep introducing it into human populations. With the COVID thing, that actually might disrupt our polio vaccination efforts because of its disruptive nature for healthcare worldwide. So unfortunately, we may see a bit of a resurgence in polio in those places because of the COVID epidemic. COVID is also an example of a virus that jumped over from an animal reservoir over into humans. And as a result, it's possible, even if we manage to vaccinate everybody and sort of get rid of COVID as we have right now, as we're experiencing it right now, there's always that possibility that a similar virus or the same virus could jump again into human populations from an animal reservoir. So when choosing viruses for potential eradication, we're pretty much restricted to those that only have, that are human specific, that don't have an animal reservoir. So immunology has been a very successful discipline. Unfortunately, in recent years, we've seen that success be undermined by anti-vax type propaganda, which unfortunately spreads a lot of misinformation. Some of it's actually specifically targeted toward communities of faith. There was an example a couple of years ago of a measles outbreak in Abbotsford in a particular Christian group because that Christian group was strongly anti-vax. They have a theology that says, you should only just pray. You should never go to a doctor or anything like that. So because you're going to learn about immunology and become more informed and skilled in the science, it's going to be up to you in some ways, I would think, to be ambassadors of this science to people in your sphere who aren't as well trained in biology. So as you learn about immunology, think about your friend group, think about your peer group, your family. Try to bring them, good information that helps them become more trusting of vaccination, helps them understand what the relative risks are, and properly prepared vaccines have an extremely low risk for them. So this is one of the things that has been the most successful public health initiatives worldwide, and unfortunately we're starting to lose some ground because of that, because of anti-vax movements. And of course the COVID-19 threw a huge wrench into that whole thing, but we'll talk about that more as we go. Okay, let's talk a little bit about microorganisms. Microorganisms are the primary cause of infectious disease. There's in fact only one example of an infectious disease that's not caused by a microorganism, and that's prions. You can see that question down. You might be familiar with prion diseases. That's the one sort of very rare exception. The germ theory of disease, if you've ever heard it called that, has been a very successful theory. So that gets a little bit into epistemology, which is how we know what we know. We call things in science theories. There's often a lot of confusion about what a theory is in science. A theory is not conjecture or guesswork or something like that. It's a well-tested explanatory framework that makes sense of the data that we have. I'm going to put up on Moodle an article I'd like you to read about scientific epistemology, how we know, what a theory is in science. I wrote it because of the evolution issue. It's very common for people to say evolution is just a theory. The Big Bang is just a theory, whatever, things like that. And when they say that, they typically mean, oh, it's not very well supported, it's just a guess, it's just conjecture. But it's not. It's actually a well-tested explanatory framework that makes a huge amount of sense of the data that we have. So what a theory is in science and how that works is something I'll put up on Moodle for you. It's well worth your time to have a look at. But that just brings us back to this germ theory of disease, the idea that communicable diseases are caused by microorganisms. We haven't always known that. Once that hypothesis was put forward, we've continually found new evidence to support that hypothesis, and over time, that hypothesis has become an explanatory framework that makes accurate predictions and has not yet been falsified. We haven't yet, shown that idea to be an error, with the one rare exception of prion diseases. So, communicable diseases are caused by microorganisms, viruses, bacteria, protozoa, invertebrates, some invertebrates like worms and things like that. So there's this vast sort of horde of potential pathogens out there. Now, most of our experience with microorganisms is actually benign. We have a lot of organisms in our systems that are commensal. So living in our gut, for example, we have a lot of commensal bacteria on the surface of our skin, a lot of commensal bacteria. And they provide actually quite a useful function for us in that they sort of take up real estate that prevents other organisms from getting a foothold in our systems. Now, those organisms, even though they're normally commensal with us, can be problematic at times if we get sick or if we take antibiotics and we kill off a bunch of them, sort of leaving real estate available for other things to move in, or for organisms that are living on us to get into places where they shouldn't get to. So sometimes you'll see opportunistic pathogens referenced, and that's saying something that is normally benign, normally commensal, suddenly getting into a place where it shouldn't be. So some of our pathogens are opportunistic. Some of our pathogens are just pathogens. When we encounter them, they cause illness for us. So the idea here is that you need to think about, there's this sort of vast potential pathogen pool out there. If you think about how quickly microorganisms replicate and how quickly they evolve, evolution being the change in average characteristics of a population over time, small organisms like microorganisms, they replicate very quickly. They have a lot of generations in a short period of time, and as a result, variation that is in those pools of different organisms can change its characteristics quite quickly over time, much more quickly than we can change on a generation by generation basis. So we have this situation where we have all of this potential, sort of pathogen pool out there, and we also have quite limited resources to deal with it. If you think about it, vertebrates only have about 20 to 25,000-ish genes or so. And if we're going to use those in some way to combat these microorganisms that are out there, then you can see that we have a very limited repertoire to draw from. And of course, most of those genes are doing other things other than immune system function. So just regular housekeeping genes or regular biochemistry to keep our cells running. So we have a very limited set of genes that we can use for immune system function, and yet we have this almost limitless evolving potential pool of pathogens out there. So one of the key questions for immunology is, how do organisms with adaptive immunity use such a small subset of their genetic material to fight off this potentially almost limitless pathogen pool? Okay, I'm getting ahead of myself a little bit. Okay, so that's sort of the us versus them thing. There's just so much out there that potentially could be pathogenic to us. We have to use our limited resources to be able to engage that and to fight it off. One of the things that we've learned in the last number of years is that our immune systems are actually useful for fighting cancer as well. Later on in the course, we're going to do one chapter that really looks at this in detail. It's a very interesting chapter. But the idea is, what happens when cells in our own body lose their ability to regulate their growth and division? The body has ways to sense when this has taken place, and it uses the immune system as a way to control and kill off those cells. So you'll see on the slide here some of the cells that are involved with that. And so that's not an us versus them kind of thing. That's an us versus malignant us. And it's interesting that our systems use similar mechanisms to fight things from the outside and also fight things that have gone wrong on the inside. So it's not always just us versus them. Sometimes it's us versus us. I've already been kind of alluding to these two questions, but this is the big framework. If we want to talk about the big picture ideas of the course, these are the two biggest ideas in the course. And as an aside, you will face these questions on your final exam. There you go. I'm going to tell you what the final exam is, and we're only like right in the first lecture here. There are the two big questions that help us frame most of the material that we cover in the course are these. The immune system has to be able to distinguish self from non-self, or in the case of cancer, healthy self versus malignant self. So that's a major, major question. It's also related to the second question. The immune system, as we've been discussing, has to have the ability to generate a huge immune response, a very diverse immune response for all these potential different pathogens that are out there. But we do it with very limited genetic resources. So how does that work? And actually, if you stop and think about it a bit, it's actually something worth pondering. I mean, some of you might already know the answer because you've read ahead in the textbook or something like that. But let's think about this. If you have a limited subset of entities to work with, how can you use that limited subset to combat something that is almost limitless in its ability to have diversity? So there's all that diversity out there. How do we develop diversity within, even if we have a limited subset to deal with? So these are the two big questions for the course. And like I said, on the final exam, I'm going to give you essay questions. These two essay questions are going to be a big part of your final exam. That's a part of this sort of big picture idea that I want you to take with you from the course. How do we distinguish self from non-self? Hugely important. How do we develop a diverse immune response? Hugely important. And how do we do that with limited resources? So those are our two big questions for the course.